Morning, everyone. So we are gathered today to attend the events of Sébastien Rigollet, who obtained the grade, the degree of Doctor of Physics, in the specialty physics for the lab science. And before handing over to Sébastien, I propose that the member of the jury briefly introduce themselves. In order to
repeat yourself.
Yes,
Two sentences. So I'm Clemens. I'm an associate professor in the UMC of Utrecht, which is the University Medical Center, and I'm head of therapeutic ultrasound.
Good morning, I'm Marlène. I'm a senior researcher in Lyon in Carmen. I work on the physics with the parametric MRI.
Good morning, I'm Cyril Lafon. I'm the head of LabTAU in Lyon. It's a lab which works on ultrasound. Very involved in delivery opening with ultrasound, and I'm Inserm research.
Yes. Hello, I'm Bridal. I'm a director of research CNRS and direct the Laboratory of Imagery Biomedical in Paris.
It's not advanced, so I will try to do my best. This is Sébastien. You have 30 to 45 minutes to present your data.
So thank you everyone in the room and online for their presence to attend my defense thesis. I'm really happy today to present my work, to present my thesis entitled Impact of the Blood-Brain Barrier Permeabilisation by Focused Ultrasound on the Brain, a quantitative MRI approach. So this thesis is part of a collaborative project between two laboratories in Grenoble and in Orléans, and the company MR-guided Therapy, which is specialized in focused ultrasound devices for therapeutic purposes. So my thesis was supervised by Drs. Emmanuel Barbier and Basile Soupar from Grenoble, and Dr. Anthony Delalande and Professor Chantal Pichon in Orléans, as well as Eric Dumont as the industrial supervisor from image-guided therapy. So from the title, several questions may arise. First, what is the blood-brain barrier? Why do we need to commemorate it? And what can be the impact of this permeabilisation on the brain? So before getting into more diving into details, let's take a step back and understand understand the broader context. So according to the World Health Organization, in 2021, they estimated that 1/3 of the global population was affected by a neurological condition, and this is the leading cause of disability worldwide. And since the last 30 years, the prevalence increased by almost 60%. So researchers try to develop different treatments from Alzheimer's disease to brain tumors. As you can see here, many treatments were developed for brain tumors, but many of them are hampered by the blood-brain barrier. So this is a protective barrier in the brain that is made of endothelial cells maintained by tight junctions, surrounded by basal lamina, neurons, astrocytic end clips, and pericytes. So this barrier is very specific to the brain, and more than 98% of small molecule drugs do not cross this blood-brain barrier. So researchers have been developing methods to overcome this barrier. Among these methods, there is some nanoparticle types. For example, direct injection method as intracranial delivery, intrathecal delivery, or intranasal delivery. Also, they developed biological mechanisms as receptor-mediated transcytosis method, or either external stimuli and even AAV variants. As you can see here, after the injection of intravenous AAV PHPD variants, the whole brain is transfused. Among these methods, almost all of these methods have advantages and drawbacks. For example, nanoparticles can be very toxic, as well as engineered viruses can induce a high immune reaction. Or even, for example, the direct injection into the brain is very invasive. Yet, the use of focused ultrasound coupled with microbubbles enables to cross the blood-brain barrier at a quite low cost and presents moderate risks and is very effective. So this method is based on ultrasounds that are focalized into the brain. Before that, we inject microbubbles into the blood system, and with the acoustic pressure, we can make those microbubbles vibrate. So we can record the activated microbubbles with a passive cavitation detector. And when these microbubbles are under a low pressure focused ultrasound beam, we can see that they vibrate gently and they produce in the spectrum foyer some harmonics. If we increase a little bit the pressure from 100 to 500 kilopascal, they start to vibrate more intensively, and we can see that some ultraharmonics as well as subharmonic frequencies appear into the spectrum, into the Fourier spectrum. And finally, if we increase too much the pressure, microbubbles start to explode and produce some fragmentations, and this is a particular this produces a particular pattern in the Fourier spectrum with a large hump in the Brodmann noise. Focused ultrasound mediated blood-brain barrier permeabilisation have broad applications as chemotherapy delivery or immunotherapy delivery in brain tumors, as well as antibodies, stem cells, and even the use of focused ultrasound to permeabilize the blood-brain barrier alone has a therapeutic effect. And among them, some of them have already been involved into in clinical trials. But the permeabilization of this blood-brain barrier is associated with secondary effects. So as the blood-brain barrier is permeable and will be intact after 24 hours, different events occur as an inflammatory response, as it was firstly published by Kovacs and colleagues. So as you can see here, some high recruitment of microglia in the permeabilized zone occurred 6 hours after the blood-brain barrier opening, as well as an associated activation. So this is a neuroinflammation marker. So many studies have analyzed different and single secondary effects, and there is a lack of consensus on focused ultrasound parameters used, and studies are very difficult to compare. So after a broad literature review, we can see that only a small amount of effects are widely studied yet, and most of them are poorly studied. Even as neuroinflammation, for example, is widely studied, yet the timeline of this neuroinflammation remains unclear, and many of this secondary effects are not studied yet. And my PhD work focused on some of these parameters or secondary effects that were or unstudied or poorly studied. While permeabilizing the blood-brain barrier, we can have benefits with blood-brain barrier permeability. We can deliver drugs. It is associated, for example, in the improvement in behavior due to increase in neuronal function, or even neuroinflammation triggered that helps brain clearance of, for example, amyloid beta plaques into the brain of mice, Alzheimer's disease mice model. But it is also associated with risks, as the blood-brain barrier permeability enables the entrance of pathogens. It's also associated with, it could be associated with important adverse events as measured in patients of leukopenia. Also, damaged BBB favors neurodegenerative processes, and the repeated cycles of neuroinflammation in the case of repeated focused ultrasound treatment is could be associated also with favoring terrain for neurodegenerative disease, as it is the case in repeated mild traumatic brain injuries in football players, published by Nicole Marquet and colleagues. So this effect needs to be closely regulated with cavitation control during a focused ultrasound procedure in order to favor the benefits and favor the benefits. So in this context, my thesis project aimed at characterize novel MR-guided focused ultrasound system at Grenoble, provided by the company MR-guided FUS, integrate a cavitation monitoring processing pipeline to control the blood-brain barrier opening safety, and also characterize FUS-induced blood-brain barrier opening vascular changes using quantitative MRI method, as well as characterize cellular bioeffects in the brain, and translate all knowledge into therapeutic protocols to improve drug delivery and rodent outcome. So I will start with a brief recapitulation of the experimental setup. Then I will present you the first study I performed on single blood-brain barrier opening, and the second study on the repeated blood-brain barrier opening. So these studies were performed on the healthy brains, and I will present to you the study on for a novel strategy for glioblastoma treatment. So due to time constraint, I will only present those three studies. So let's start with the experimental setup. So every experiments were performed on the image facility with a 4.7 Tesla scanner and the image-guided therapy device, which is which embeds an imaging transducer with 7 emitters and 1 receiver. And with this device, we can focalize the ultrasonic beam with the guidance of the MRI into different brain regions, as for example the cortex or the striatum, and precisely control the location of the energy deposition. The transducer was characterized using a 3-axis motorized system and a hydrophone. And as you can see, we modeled a rice grain shaped focused ultrasound beam, as well as a linear relationship between the peak negative pressure and the amplitude of our ultrasonic pulses. And this peak negative pressure was correcting each time regarding the depth of focalization and also the skull attenuation of the rodents. Finally, we made microbubbles to open the blood-brain barrier, and we use microbubbles made at Orléans, the laboratory of Professor Chantal Pichon. So those microbubbles were produced by mechanical agitation with a vial mix and with a lipidic shell. They encapsulate a perfluorobutane gas, and they are stabilized with polyethylene glycols. Those microbubbles have an average diameter of 1.37 micrometers, so they are suitable for focused ultrasound blood-brain barrier opening method. So with all of this method, we now I will use them for the following studies. So the first study, we looked at single blood-brain barrier opening modification into the brain. So I talked to you about the secondary bioeffects in the brain, and if we specifically look at vascular changes, Raymond and colleagues measured in 2007 a transient vasoconstriction with biphoton imaging following the application of focused ultrasound, as well as Fan and colleagues measured right after the application of focused ultrasound a large drop of perfusion, but this blood-brain barrier opening was hemorrhagic, so unsafe. More recently, at the lab, Vasily Stupar and colleagues measured after the application of focused ultrasound large drop of perfusion, and this was confirmed lately, very recently, by Labridge and colleagues. As you can see here, after the permeabilisation of the right hemisphere, we have a drop of perfusion also in that can be seen in the cortex and here the striatum. But several limitations were found from all of these studies, like the field of view was limited with biphoton imaging, the blood-brain barrier opening was not reversible by Fan and colleagues, as well as some memory methods were not accurate, as in the study of Todd and colleagues, and studies led by Stupar and Labridge were used a large focused ultrasound beam, so some large vessels could be involved into the drop of perfusion. So no studies have evaluated vascular changes with together with cavitation, and also what are the potential cellular effects associated. So for this experiment, we used the Wiart on rats. We permeate the blood-brain barrier with pulses of 50 milliseconds every second for 1 minute, and we used 600 kilopascal pressure, and measured quantitative MRI parameters, so the following ones: the apparent diffusion coefficient of water, the cerebral blood flow, the blood volume fraction, and the blood-brain barrier permeability. So we divided our study into 3 animal groups. The first one aimed at measuring the cerebral blood flow 30 minutes following the permeabilization of the blood-brain barrier. The second group to measure the dynamic of this cerebral blood flow drop. So we measured at the CBF at 30 minutes, 60 minutes, and 90 minutes. And then the last group, we measured the blood volume fraction at 30 minutes, as the blood volume, the CBF is equal to the product between the volume and the speed. So we wanted to see where does this drop of perfusion comes from. And we couldn't perform all of this experiment at the same time, because in the first we needed to inject different contrast agents. As in the last group, we used iron-like contrast agents to measure the blood volume fraction. That could not be associated or done while we are measuring the cerebral blood flow. And from group 1 and 2, we permeabilize the blood-brain barrier either or in the cortex or in the striatum to see the differential effects between permeabilization in these 2 regions. We monitor the cavitation with the passive cavitation detector. We windowed our signal onto 200 microsecond windows and performed the Fourier transform. We then used, in order to characterize cavitation, the formula recently published by Novel and Clemens. And so, as you can see here, we represent the exposure time, and each line is one ultrasonic pulse. So pulses are 50 milliseconds long. And if we take one pulse, we can see that we computed the initial, so the intrapulse ultra-harmonic dose and intrapulse harmonic dose. and we define a threshold at 1.5 decibel to determine if there is inertial cavitation event. So after performing the blood-brain barrier permeabilisation, we divided in 3 groups or animals. So we characterize blood-brain barrier opening as soft, mild, or hard, depending on the inertial cavitation events. So as you can see, for mild group, we have a large amount of inertial events, as well as consecutive inertial events. We found a good correlation between those 2 parameters. So we discriminated with this index for animals in order to remove the animals that underwent too strong blood-brain barrier opening. And in our study, only 1 animal had strong adverse effects, as you can see on this micrograph here. So 30 minutes after the blood-brain barrier permeabilisation, we can see we permeabilize in this group the cortex and here the striatum. We can see a large drop of perfusion that was found significant, and 24 hours we did not find any difference. So as you can see, there is a big mismatch between the area permeabilized and also the drop of perfusion region. And this was found either in the cortex and in the striatum. Then we measured at also 60 and 90 minutes the perfusion in brains. And as you can see, it starts to return to baseline values. And so this phenomenon is transient and lasts less than 2 hours. Regarding the blood volume fraction parameter, also we permeabilized the blood-brain barrier with a single spot, and we measured a large drop of blood volume fraction in the whole cortex. And this value was found also. We found a significant decrease in the cortex. For the permeabilization of the striatum, we also measured a decrease, yet we didn't find any significant difference, very probably due to the low number of Now, regarding the neuroinflammation, we harvested brains between 1 day to 7 days after the permeabilization of the blood-brain barrier, and performed the histological staining with GFAP marker, that is a marker of neuroinflammation. And we found the peak intensity of this marker 2 days after the permeabilization of the blood-brain barrier. Regarding the microglia, we found an increase, a significant increase in microglia recruitment only 1 day following the permeabilization of the blood-brain barrier. That was expected because microglia are the first immune cells that react after a brain aggression. So in this study, we monitored the cavitation and proposed to classify the animals in order to remove those who received too intense treatments. We measured the transient decrease of perfusion that lasted less than 2 hours, and this was associated with a reduction in blood volume fraction. Yet, there was a big mismatch between the amount of drop of perfusion and the drop of blood volume fraction. So the reduction in volume does not explain fully how the perfusion decreased. We also measured the transient neuroinflammation with astrocytic and microglia activation. And also the reduction in cerebral blood flow and blood volume fraction are still lower than what we can find in a stroke model during the occlusion. Respectively, 70% of perfusion and 50% of volume fraction was measured by former colleagues here with the same MR method. So this mild ischemia that could occur could be balanced with hyperoxygenation of the animals just after the focus ultrasound to prevent this mild ischemia. And the phenomenon could originate from either cortical spreading diffusion mechanism or a vasospasm that was already, we know that this can occur during the permeabilization of the blood vessel. Now, if we use repeated focus ultrasound to treat, for example, brain tumor patients, we could imagine that the phenomenon already measured after a single blood-brain barrier opening could be even stronger. So this is what we've done for the second study, and we use MRI fingerprinting to monitor those vascular changes. So in this study, we use the same animals. We use a larger raster scan in order to permeabilize repeatedly at the same area, in the same area as the blood-brain barrier. We use two different pressures to see the impact of the pressure on the repetition of the procedure, so 390 kilopascal and 440 kilopascal. And we measured the same parameters as in the first study, and thanks to the MRI fingerprinting, we could measure also the oxygenation of the tissue and the mean vessel radius. We divided in this study the animals in three groups. First, a single group that received only one treatment, then a group that received 4 treatments, so 1 treatment a week, and a group that received 2 treatments a week for 4 weeks, and harvested the brain at the end. Regarding the results, we can see that the permeabilization of the blood-brain barrier was highly reproducible. As you can see here in black, this region was permeabilized 8 times, and in light yellow only once. And we can see we have the similar pattern as our raster scan. So blood-brain barrier opening was reproducible. Also, we did not find any edema or microhemorrhages on T280 MRI and after histological investigation. So regarding the blood-brain barrier permeability, we measured how fast the contrast agent passes the blood-brain barrier, and we found that this time was reduced following 4 permeabilization of the blood-brain barrier at 390 kilopascal and 440 kilopascal when this permeabilization was performed twice a week. And this was not found different in the other groups. So we measured the slightly altered blood-brain barrier permeability following the application of 2 treatments a week for 4 weeks. Regarding the cerebral blood flow modifications, we measured, as in the study 1, a large drop of perfusion in the ipsilateral region compared to contralateral. And this region was also restrained to the area permeabilized. Regarding the groups that received 4 or 8 treatments, we also measured important decrease in cerebral blood flow in the permeabilized regions. Yet, we did not find any increase in the effect. So there was no impact of the repeated treatment on the cerebral blood flow drop. As we measured a large drop of perfusion, we could imagine that there is also a large drop of oxygenation. So we measured the oxygenation in the tissue following either 1, 4, or 8 treatments. So we only present the results for the groups that received the highest pressure. And surprisingly, we did not measure a large drop of oxygenation, only between 2 to 8% decrease in SO2 values compared to contralateral. So regarding the inflammation, we harvested the brain 1 week following after the last treatment. And as you can see, we measured some larger vessels in the permeabilized region, only for the group that received the highest pressure at 8 treatments. Also, we measured the high astrocytic recruitment for this group, and we did not find any difference for the other groups. So the effect of the repeated treatment twice a week at the highest pressure is stronger than the single application of focused ultrasound, and also the application of one treatment a week. So a little summary of the results. So we did not measure any differences into the diffusion water parameter. We measured the similar drop of perfusion, even if there was a slight increase in this drop for the group that received the strongest treatment. For the parameters of blood volume fraction and mean vessel radius, we surprisingly measured higher values in the ipsilateral region compared to contralateral region. And this was confirmed 1 week later with increase in vessel diameter, and also a significant increase of the astrocyte recruitment 1 week after. So if we compare the studies 1 and 2, we measured a similar drop of perfusion. So enlarging the treatment area does not appear to increase the effect of reducing perfusion. We measured very surprisingly, we measured the drop of blood volume fraction after a single application of focused ultrasound, and an increase in the blood volume fraction values for the group that received large blood-brain barrier opening. As we could imagine that the vessels were lose their contractility, for example. And also we did not measure any increase in vessel diameter for studying the study 1. And 1 week after also did not measure any significant difference. So from this second study, what we can conclude is that using only one treatment a week would be better, as 2 treatments a week at high pressure, quite high pressure, induces important changes. So now, regarding our experience, we wanted to see how our focused ultrasound sequences could improve the delivery of therapeutics, and we used our experience to test a novel strategy for glioblastoma treatment. So as you may know, glioblastoma is the most common primary disease in the adult brain, and its incidence is in constant increase. The standard treatment involves the surgical resection of the tumor core, and as stated also is they use chemotherapy and radiotherapy. So as you can see in this image, the glioblastoma is divided into 2 regions. The first one, the tumor core, and around the peritumoral brain zone. So both of these regions have different features. For example, in the tumor core, we can achieve high drug concentration, yet there is also a high drug clearance. And in the peritumoral brain zone, there is an intact blood-brain barrier. Thus, we cannot. There is a low drug penetration, and this is the part to treat. So, with a collaborative work with the Strobe Lab and Dr. Emmanuel, that had the experience into the F98 rat model, they found 10 years ago that after intracerebral delivery of carboplatin associated with radiotherapy, they demonstrated unprecedented survival and improvement. Yet this technique was very invasive. And more recently, a study by McDonald and colleagues demonstrated that the application of focused ultrasound associated with an injection of carboplatin improved significantly the survival outcome. So they permeabilized the blood-brain barrier twice and measured a significant improvement. So we wondered if we can use our expertise on focused ultrasound to improve the delivery of carboplatin and perform radiotherapy in the brain in this F98 rat model. So here is the experimental timeline. So we injected firstly 1000 F98 cells in the rat striatum. 14 days after, we permeabilized the blood-brain barrier with a microbubble and injected carboplatin into the vascular system. We used a raster scan of 8 by times 8 square millimeters at a pressure of 470 kilopascals. That has the same mechanical index as the study of McDonald's and colleagues to be to compare both studies. And we divided the groups in two, with the first group to study the biodistribution of the chemotherapy, and a survival group that received a single radiotherapy session of 15 gray around the brain tumor. So regarding the results of the biodistribution, we measured the platinum concentration using inductively coupled plasma mass spectroscopy at Département de Pharmacochimie moléculaire. And we measured with the permeabilization of the blood-brain barrier an important increase into the concentration of the platinum, either in the tumor core and also in the peritumoral brain region, the area that needs to be treated. Also, 24 hours after, we found still a large increase in platinum concentrations compared to the group that did not receive any focused ultrasound. So this led in the survival study to a large increase in the median survival time. So the full treated group compared to untreated group led to an increase in median survival time of 158%. And compared to the radiotherapy group, we measured a 52% increase in the median survival time. Also, this was associated with a larger reduction in tumor volume progression. As you can see here, at 21 days following the treatment, the tumor core was found less important than the radiotherapy group. So to conclude on this last study, the treatment combination was safe, decreased the tumor progression, and improved the median survival time by more than 50% compared to the radiotherapy group. And focused ultrasound blood-brain barrier opening enhanced the carboplatin concentration by a factor 3 in the tumor core and by a factor 4 in the peritumoral brain zone. If we compare our study to others, we can see that this study is demonstrated the best results using non-invasive methods up to now. And using repeated treatments could improve greatly the RAP outcome. So this will be the next study, hopefully. And we could also clinically transfer this protocol in the following years, as all of the focused ultrasound procedure was validated in glioblastoma patients, as well as chemotherapy and radiotherapy is already available in clinic. So now, if we sum up this whole thesis project, the main findings were that cavitation monitoring pipeline should be performed to represent what could be done in clinic and avoid too intense energy deposition. And also we measured with quantitative MRI for the first time a large number of physiological parameters, as well as important vascular alteration that were measured in absence of lesion, as hemorrhages, for example. And we evaluated cellular aspects related to neuroinflammation that could be triggered by these vascular alterations. We also used different effective and safe focus ultrasound pattern for rodents using different pressure, size of broadband biovolume, either using a single or repeated focus ultrasound treatment. And the clinical perspective of this work are the measure of the perfusion and brain oxygenation following focus ultrasound that should be performed with MRI as soon as possible. Also, inflammation monitoring with PET scan could be of great interest in order to see if we have the same pattern or if we have a strong neuroinflammation following the procedure, as well as secondary bioeffects need to be further investigated, even at long term. And future developments needs to be made in order to control this regarding either the focus ultrasound itself, the calibration control, and the engineering of the microdoses. Along this last 3 years, I had a great opportunity to attend many international conferences, as well as published first article in the International Journal. I was also involved in the supervision of 4 interns during the last 3 years that did a really great job. And with this, I want to thank, of course, my thesis supervisors, the MR-gFUS team, the team from Orléans for the microbubbles, of course, the wonderful interns I supervise, as well as image-guided therapy for the support, the stroke tip for the glioblastoma study, and of course the jury members for their presence. And I will be very happy to answer any question. Thank you.